All right. Hello, everyone. So we're back at another episode of Sensibly Curious About Vaccines. And I'm Tracy Høeg, and this is my co-host, Christine Stabell Benn. And we have two guests today that I'm so excited about. And I'm going to introduce them. I'll probably start with Retsef because I met Retsef in person. I guess about a year and a half ago.
Yeah, something
2
like
years
that.
ago, almost 2 years ago, in Washington, D.C. at the Academy of Science and Freedom at Hillsdale College conference. And so Retsef, I should probably introduce the episode by actually saying that we're going to be talking about a pretty hot topic at the moment, which is how we're going to be specifically looking at the Pfizer vaccine and how it was tested in the original clinical trial using something called Process 1, so manufacturing practices, and then how at the very end of that clinical trial, the manufacturing process got switched to Process 2, and that was not actually thoroughly tested. It was not tested in more than 252 subjects out of the over 21,000 that initially received the vaccine in the original Pfizer clinical trial. And we're going to talk about what that means about sort of what we do and don't know about the safety and efficacy of the Pfizer vaccine based on the limited data that we got from the initial clinical trial. And then I think we're going to be talking about like how this happened, why it happened, what Pfizer knew when, what the EMA and FDA and our regulators knew when, and what it means for us, for people who are vaccinated, for the people who we know who are vaccinated, for the future of vaccine safety. So to get back to introducing our guests, so Retsef Levi, so I hope I said that right. So he is originally from Israel, as far as I understand, and he originally studied mathematics at Tel Aviv University, and then went to Cornell to study research operations, and now he's a professor. Operations research, sorry. And then you're a professor of operations management at MIT right now.
Mm-hmm.
And I think actually, if I remember correctly, you got interested in studying vaccines after COVID started. Is that correct? Is that kind of when you got, or was it before?
So I was actually actively studying the issues related to safety and quality of manufacturing of biologic drugs, or biologic drugs more generally speaking, and I've been having multiple contracts and awards from the FDA, still do, and also more generally speaking, my research has a lot to do with the intersection of data and analytics and advanced analytics, and how you use that to inform the design and operations of complex systems. So I've been working a lot with hospitals and health systems. So basically, almost from the start of my academic career, I've been looking on health-related data from large health systems, from manufacturing environments of biologic drugs. And of course, when COVID started, it actually was related not only to the clinical aspects or the health aspects of my research, because other parts of my research are about logistic operations, risk management, which had a lot of aspects relevant to that as well, in terms of how you manage the pandemic risks, and how do you protect, for example, nursing homes and things like that. But definitely over the last 3 years, I've learned much more about biology and immunology and other aspects that maybe I didn't know about a whole lot about before. But to some extent, operations research is exactly about the ability to use analytical tools and modeling tools in different domains. And you usually work with the subject matter experts, and you learn the topics, and you bring the perspective of data and modeling analytics to assess risk, to understand how you can optimize systems and so forth. So that's kind of my angle where I'm coming to these topics. So.
Got it. Yes. And I should say that listeners will actually probably know you from maybe best from your research looking at ambulance calls in Israel related to the vaccine rollout. I don't know, do you want to, and cardiac deaths as well related to the vaccine rollout. Do you
Yeah,
want to
so
just
more,
quickly
yeah.
talk?
Yeah, so once the issues related to myocarditis and pericarditis emerge in the context of the COVID-19 vaccines, but not only, also in the context of the COVID illness, I started to read about this, and one of the first things that you learn about myocarditis is that it has a lot of subclinical cases on top of the clinical cases, and that also it's a known cause of sudden cardiac death out of hospitals, especially among young people. And when I started reading about this, I said, okay, so if that's the case, these are signals that the current surveillance systems are not likely to detect because it's out of the hospital. So many of these cases will happen in settings that may not be kind of easily detectable, and then kind of the idea of, okay, let's just look on what happens with EMS calls. And I was able to connect with senior VP for research in the national EMS organization in Israel. Israel is a country where there's only one EMS, unlike the US, where it's city by city. And we started to look on the temporal patterns over a course of 2 years, and we indeed saw a major increase among young people 16 to 39 of the cardiac arrest EMS calls. When we looked on the temporal correlations, when we compared it, because we don't have individual data, unfortunately, we only have aggregated data, we were able to detect the temporal correlation with the number of doses that were given to these ages. And we didn't see the same correlation for COVID-19 infections, which, to be honest, I don't think our paper, the major contribution of our paper was to raise the signal that there is a correlation that needs to be checked more thoroughly and more kind of carefully to see whether that is a plausible risk. I think since then, although the paper was attacked viciously by people that didn't like even the idea that there is potential risk, but now by now we know that this is not a theoretical scenario. There are multiple papers, including autopsy-based, that show that it is a real risk. But unfortunately, still in Israel and other places, I don't think that people were able to basically bring together EMS calls with individual level data on vaccination status and infection status to do a thorough job on understanding that type of risk. But to me, it kind of showed that when we look on surveillance of in the context of safety of drugs, we need to incorporate all sources of data, including what is happening out of the hospital, because that's kind of these are incidents that are not likely to be detected by the regular surveillance systems that we currently have.
Retsef, am I right that you took your findings to people at the Ministry of Health?
Yeah, and also the FDA as well. So
and what was their reaction?
The reaction was that they don't see any increase whatsoever in cardiac related events, which by now we know it's not true. In fact, in another data that I still did not write the paper on, but we analyzed it, we received from the two HMOs, the large HMOs. In Israel, the healthcare system is organized through multiple HMO, healthcare maintenance organizations that basically are responsible for longitude care of decades of a large population. So we got from two HMOs that are responsible for over 6 million people in Israel, data about pulmonary embolism cases, diagnosis. And yet again, you see a major increase in 2021 in all age groups of this diagnosis. This is actually including the clinic diagnosis, not out of the hospital diagnosis. But the point is, again, on pulmonary embolism, that it can cause sudden death. In fact, sometimes it's being confused with cardiac arrest. It's hard to distinguish them. And we see the increase, including in ages 0 to 29, major increase, like 17%. So I think that the Ministry of Health in Israel was not very forthcoming, to say the least, to do this investigation. In fact, they declared in social media that they are going to try to cause our paper to be retracted. So they even threaten us in social media that that's what they're going to do. So, you know, I'm pretty sure that by now it's not that surprising, the story that I tell now about the reaction to what we found. Unfortunately, I think that once healthcare regulatory agencies decided to give this vaccine to all ages, including young people that are not high risk, they committed themselves to a path that they cannot admit any wrong. And I think that we see the reason why we see this great level of denial and efforts to suppress any attempt to understand what the risks are, in spite of mounting signals, is because they are so committed. Like, what are they going to say now if there is a real signal that will harm or potentially harm young individuals or children that have zero risk from COVID? It's going to be very hard to rationalize this. I think if they just were limited to the high risk population, even if retrospectively that would have turned out to be not the best decision, it would be a legitimate decision, because under the uncertainty at the end of 2020, you know, that would be reasonable to make a decision like that. But unfortunately, they didn't do that. And I think that that's kind of the key reason why I think we are facing a lot of the resistance to investigate anything.
I think that's so important, and one of the reasons that I think podcasts like this are so important too is to, you know, talk about our experience as researchers. And, you know, now, like you correctly stated, your findings have been validated in different ways by different research groups, like by the autopsy findings in the Korean study, and by the work in the UK, the epidemiological work, the self-controlled case series that they did, finding increased risk of cardiac death in young males as well. And you found it in males and females. And I would say, like, the Nordic study found a higher risk of myocarditis after COVID, or after, sorry, the COVID vaccines than after COVID-19. By a lot, in males and females who are young, 16 to 24, and they basically found in young people, especially in children, they couldn't find any COVID myocarditis. And so it's interesting how, you know, I feel like multiple research groups, you know, ended up finding a similar thing, but I'm sure that there were there have also been many groups that haven't been able to get their findings published,
yes,
and they faced pushback. That you did, and
let me put it this way:
well.
the sagas around the paper that we just talked about could be a podcast by itself. But just to add on what you said, there are also two prospective studies in which people tested the hearts of young people that got the vaccines, one from Thailand, one from Switzerland, and in which you see 2-3%, which is a dramatic number of people that have increased troponin, right? Like, I mean, even if not subclinical, like increased troponin, which is an indication of heart muscle damage. And there's even a more recent study in which they actually find the mRNA in the hearts of people that died shortly after the vaccine. So I always say, and maybe here I'm bringing my intelligence hat, because I spent in Israel before coming to academia. I spent almost 12 years in intelligence. Often we tend to see, oh, this indication maybe doesn't prove it, and that indication does, or that finding. But you have to step back at some point and look on the mounting evidence and just like say, hey, something is wrong here. It cannot be that we see so many signals that all speaks to they all speak to the same kind of direction, that there is a major cardiovascular impact, especially on young people. And you have to ask yourself very tough questions. Why would you want to take such a risk where the risk from the illness is clearly, I mean, I don't want to say zero, but it's very close to zero, especially with the current variants. So
yeah, well, and I think it's good that we introduce ourselves too like this, because I think it's important to know we're just all like not conspiracy theorists. We're, you know, all researchers, scientists. I was understanding from Josh, I think we may even have sort of a similar background in a way. I don't know, but well, so you're originally from California. And you so you studied sociology. You studied, let me get this right. So you went to UC Berkeley for your undergrad, and you studied sociology, and then you went to Princeton for your PhD. And now you're at Hebrew University. And I kind of said we have a similar background because I'm from the US as well, and I actually think we like graduate college the same year. And
Oh,
and then I ended up moving to Denmark.
To where?
To Denmark.
Oh, okay.
After I did my MD, I did my PhD at the University of Copenhagen.
Oh, wow. Okay.
And so like I think we both have the experience of like growing up in the United States and then moving abroad, and studying in two different countries. And I think that actually brings a lot of critical thinking skills. And I think Retsef sort of has the same, but like the opposite. He started in Israel and moved to the United States. And I think once you see that, you know, different countries kind of do things very different ways and have different what's the word, they have different customs and things that they take for granted that they just do without thinking about them. And that was definitely the case when I moved to Denmark, that so many things in medicine were done differently, such as I've been talking about with Christine a lot, that like the vaccines that were given in Denmark were like just, you know, about a third of what was given in the United States. And just that alone made me pause, as well as the fact that there are no vaccine mandates. And so I'm just bringing up one example, but there are so many examples. And so it makes you kind of take a step back and think, wait a minute, you know, are the things that we do really based in science?
Right.
Or are we just doing them because people around us do them? But so I'm talking too much, but I would love to hear, Josh, like how did you get interested in studying vaccines? And I should say you're a lecturer now at Hebrew University in criminology, right?
Criminology and sociology. Yes, that's correct.
And sociology, okay.
which is
Yeah.
equivalent to a professor.
So tell us
equivalent to a professor at
I'm an associate professor. Yeah.
Yeah.
That's the corollary to the US.
That's right, associate professor.
And
yeah, the Israeli names are slightly different. So
right.
And your name is Joshua Guetzkow. I didn't even say that yet. That's important to get in there.
That's fine. And if you want to talk about my bona fides, you skipped over the postdoc I did at Harvard in health policy.
That's right.
And you know, and so people don't really understand what sociology is or what sociologists do, but you know, I have advanced statistical training at the Demography Institute, the Office of Population Research at Princeton, and the Woodrow Wilson School of Public Affairs, and in the Department of Sociology. And you know, all of these methods are pretty much similar across domains. So I have a good understanding of research methodology and statistics. So I got interested in this actually in 2019 when are you guys familiar with the book Turtles All the Way Down? You heard this book? I've heard of it. I haven't read it. This was a book that was written this was a book that was written in Hebrew, actually. I think in 2017, and the authors of the book, it was written anonymously. Okay, now you read the book, you realize that these are people who seem to have some kind of background in medicine or something like that. These are, you know, qualified people, but why would you have to feel the need to write this book anonymously. And the reason is because they were worried for their careers and the pushback that they would get, because this is a book very critical of vaccine safety. And criminology, two colleagues of mine in criminology wrote a book review of this that was published in the Journal of the Israeli Medical Association, okay, in their book review section. And basically talking about, isn't, sort of from a criminological perspective, how is it possible that people will feel afraid, to publish something that's scientific, based on scientific claims, using scientific evidence? And interestingly, and so they got interested in looking at this more broadly. And this is a phenomenon that we see, and Retsef was just talking about this, the way in which many papers that raise safety concerns about vaccines are retracted, and there are people who raise concerns about vaccines can often be face negative repercussions. And so we actually started a research project in 2019 to look at this, and that basically involved interviewing scientists and doctors who had experienced this phenomenon. And we've had a couple of papers published out of that. So that's kind of how I got interested in this. And ironically, this review that they wrote was retracted from that journal. The senior editor of that journal is one of the world's leading immunologists and experts on autoimmune diseases, Professor Schoenfeld. And Jutta Schoenfeld, and actually Science Magazine wrote a whole expose about the scandal, castigating Dr. Schoenfeld for his intransigence related to, possibly raising concerns about the safety of vaccines. So that's sort of where I entered into this. And then, as the COVID vaccines rolled out, I started looking at pharmacovigilance data, which was immediately very early on, which was very concerning, and so I wondered, well, why, how come I can use their methodology to find safety signals and they're not finding any safety signals? So then we FOIA'd the FDA, the CDC, to get their safety signal analyses. 2022 comes around, they finally answer us, and it turns out they didn't do their analysis. They didn't do the promised safety signal analysis that they were going to do of their own very own pharmacovigilance system, which is shocking in and of itself. And then it turns out that later, after they told us they didn't do it, they started doing it. And end of 2022 comes around, we finally get access to that, and it turns out they did. They found hundreds of safety signals, most of them stronger than the signal they have they found for myocarditis. And yet it's never been in the news. The CDC has never announced it publicly. And it's a real scandal. Anyway, but that's not what we're here to talk about.
No, it's actually a really interesting topic, because I mean, the CDC has been, really behind on reporting safety signals to the public. And I know that because I was the lead author on a study about the myocarditis and, post-vaccination myocarditis in adolescents. And, we came out with a much more reasonable estimate of how often it occurs in adolescents than they had come out with in the summer of 2021, only to have it validated by many international databases later. And and so they really lagged behind on letting the public know about safety signals.
Oh yeah.
And so I'm curious, where was this published that they this do you know where they ended up was it in their MMWR?
They never, no,
only Josh wrote about it.
FOIA.
Because I was going to say I haven't seen it.
It was obtained by a FOIA request from a journalist at the Epoch Times named Zach Stieber. So he obtained that. All we have are their Excel files from the CDC, and then you could just look at it and you can see what the safety signals are. So I wrote about it on my blog, but so it just hasn't.
If you send us the link, we can link to that in connection with the podcast and things.
Okay.
So
but that's another
Times article.
Yes, there is also an Epoch, but this is another example where you would expect that even if they did not write about this, Josh will be able to publish this, but it's only a Substack at the moment, or maybe a news article. But I cannot hold a little bit of cynicism, because I just before we started, I read a new analysis, a new paper. It's still not peer reviewed, but it's in the Med Archive, actually written by FDA people, that actually studies like 4 major databases of health insurance programs, and basically detects myocarditis and pericarditis as signals, but also seizures and as a potential safety signal of seizures among actually young children, 2 to 5 and 2 to 4. But the cynical aspect is that they talk about near real time surveillance, which I'm saying, oh, we are 2023. The vaccine is out there from early 2021. That doesn't look to me like near real time. So whatever you found, it's not near real time, and maybe that's kind of summarizes where we are. So,
but at least it was published by the FDA. Then,
well, slowly we are uncovering the. But I think these 2 stories, I think, just tell us that it's almost like you would expect that if you rush the approval process, which we did, and maybe for good reasons in this case, right? You would actually have a far more stringent and rapid surveillance system and put in place. But what you got is quite the opposite of that. So that's kind of striking to me. How in spite of the fact that it was very clear that we are rushing it and it's not going to go through, and it did not go through the regular timeline and the regular scrutiny during the approval process, and it was approved under emergency use authorization. I think that it was an obligation to put together the most stringent, rapid surveillance system for safety, and we see anything but that. Perhaps quite the opposite.
I absolutely agree, and I think that many things could have been done during the rollout, actually, clever rollout that had permitted unbiased comparisons of vaccinated and unvaccinated groups in the among non-high risk populations. We could have done step-wedged rollout. We could have done even randomized rollout to study overall health outcomes among vaccinated and unvaccinated. So I agree that's a great omission. But maybe we should also take that as a stepping stone to move to the subject of today's talk, which is, among others, I think we've covered a lot of interesting subjects already, but also to talk about whether there were some oversights in the approval of the Pfizer vaccine in terms of how several aspects, as I understand it, most importantly, how the actual product was made, but also later a change in buffer, how that was handled in the regulatory process. So maybe Josh, you will.
I was going to say, like, a good segue, because we're talking about the post-marketing surveillance, and to get into, what happened with this is, what we can say, what the medical journals, they're their own businesses. They sell a product. Like, we actually can't really depend on the medical journals to publish unbiased information, and they don't, because they're actually, not a public service. Whereas we count on the FDA and CDC to do that. But the FDA, we know that they're supposed to be doing post-marketing surveillance. And so a good segue to this topic is the fact that they had said, beyond that they were going to do the post-marketing surveillance on subclinical myocarditis and on the Pfizer's pregnancy trial, like that they were also going to test all of the lots with the 250 subjects who had received the process 2 vaccine. So they were going to test each of these 250 people in each of these lots. And then they just decided that it wasn't necessary. They weren't going to make Pfizer do this. And so I think maybe that's a good segue to talk about what happened. And I think I agree with Christine. I think Josh should get into exactly what happened during the trials. So to bring people up to speed who maybe don't know about the process 1 and process 2 manufacturing, and then how it maybe could have been studied, but really wasn't.
Okay, sure. So as part of the vaccine development, now here we're talking about the Pfizer vaccine only. So it was the first vaccine product was made using one particular manufacturing process. It's very small scale. You get like, I don't know, 0.7 milliliters per batch or something like that, which wasn't enough, of course, if you wanted to roll out the product and inject it into billions of arms around the world. And so they developed simultaneously to starting the trials, they were developing a new manufacturing process to upscale the production of the vaccine. And so the trial itself, so the small scale is they call it process 1, the upscale, they call it process 2. The trials were run on process 1. And then subsequent, well into the trial, which began in late July, they said, so in early October, they revised their protocol, and they said, okay, we're going to test process 2 doses on 250 people per lot, and not just people aged 16 to 55. So they had never intended to test this in the older age group. And they said, we're going to test the immunogenicity and the safety. We're going to do that by comparing those 250 people or 250 people per lot to some randomly selected 250 people who got the clinical doses, the process 1 doses.
Can I interrupt just briefly,
yeah, sure.
Just because that sounds like a huge commitment, actually. It's kind of, it didn't have any expiry date. So they basically, just to see if I understood it correctly, they said, we have vaccinated these 20-something thousand in the in the regulatory trials, in the phase 3 trials, with the vaccine, the Pfizer vaccine produced by process 1. And now we have started producing it with this process 2. But we say that for each batch of the process 2 vaccines, we'll vaccinate 250 people in a control setting to compare with these historical phase 3 participants who got process 1 vaccines.
Right.
I mean, just if it didn't come with any expiry date, was that going to go on forever, or
no? They didn't. Yeah, so it's unspecified in the protocol. They don't say how many lots. They don't say how long it's going to take. It seems to be an add-on to the phase 3 trial. So it's something that they were, and we have all the data now from the trial, and so we can look and see who got the process 2 doses, and we see that there were, in fact, they only tested 1 lot of process 2 on 252 people. And there are multiple ways that we know this. This has been confirmed by independent researchers using a different method to identify who got the vaccine dose, the process 2 lots, because one of the things the data doesn't have is what batch did this individual, what lot was this person given. That seems to be closely held.
But just to clarify, Christine, this was done while they were still recruiting people to the trial. So most of the people were recruited and got process 1, and on the tail of the recruitment, they decided to basically inject this new process 2 kind of batch.
Right. They tacked it on because
the idea was it was just during the phase 3 trials that they would test any subsequent lot. It wasn't like it would go on into 2023.
I don't think there was any intention or indication. I think the regulators knew about this switch, and they were concerned about it. And I think they did this to help ease, perhaps per the request of the regulators or something to that effect. So that at least they could say that it was tested. Now they told the regulators, we're starting this on October 19th. We know this through documents that were part of a leak or email hack of the EMA, but they actually, now we know that now we know this through a FOIA request too, but they told the regulators that they were going to start this study on October 19th, 2020, and the data would not be ready for analysis prior to the emergency authorizations that were given by many regulators around the world. They did say that they expected to have the results in January or February 2021. We now know, again, through a FOIA request of the MHRA, that they never did that comparison study. And they actually revised their protocol in fall of 2022 to say, we didn't do this, we don't need to do this now that the vaccine has been given to so many people, and therefore we can see that it's safe. We don't need to bother with doing this comparison. I mean, the comparison, frankly, is somewhat bogus anyway, because 250 people is not enough of a sample to be able to test the safety of anything. And in terms of the immunogenicity, they only took, so immunogenicity is, well, what is the antibody response to the vaccine? They only tested that on 4 people, and they didn't look at their antibody levels before they got the vaccine. They only looked for a month after the vaccine, and they only found an antibody response in 3 of the 4, all of whom were under the age of 23.
I heard you say this before, Josh, and I'm so puzzled. How could they vaccinate 250 people that was a kind of comparison group that was vaccinated with this new process 2 vaccine and not test antibodies? That seems to be the whole, I absolutely agree the numbers are too small to look at safety, but at least antibodies would be something that was straightforward, easy, cheap to measure. How do we have any idea why they didn't test this?
Well, I don't think, frankly, I think this study was kind of shambolic to begin with. It was just sort of to check a box. I don't think they ever really intended it to be meaningful. Obviously they didn't, because they never did the study that they said they were going to do. So they did do a series of comparisons between process 1 and process 2 doses. They called them analytical comparisons, and these are just different tests that they ran on the different products to see how comparable they were. And one of those tests was in vitro expression of spike protein, and they found that the levels when they do it in a vial, in a glass vial, you see that there's a similar expression of spike protein. So I guess their assumption was, well, if there is a similar expression of spike protein, there would be a similar antibody response.
But in these analytical,
sorry, go ahead.
In these analytical tests, they also saw a number of differences, and I guess we're going to get into those also in terms of the DNA content and the endotoxin levels, etc. But yeah, let's you had something.
But just to underline, I want to underline a very important fact that the emergency use authorization, in which we decided to give it to billions of people throughout the world, that decision was made with essentially minimal, basically no clinical data from patients that new process was tried on. So that analysis was not available even. The data was not even available, even theoretically speaking, because most of, we just started in October 19, and the cutoff for the EUA was less than a month later. So the fact of the matter is that we actually approved this product to use at scale with essentially no clinical data from the real manufacturing process. And I think that it's important to understand that this is fundamentally against any regulatory standard, because especially because the new process 2 is a biological manufacturing process in which you actually use live cells to scale the product, or you use biological cells to host cells to scale the product. And it's known that unlike maybe traditional drugs, when it's a chemical formula, the level of risks that you can, or the safety issues and the efficacy issues are far more complex, and so you just don't do that. It's really has to be an inherent part of the approval process, which it wasn't here. So.
And we learned the hard way with the Pandemrix vaccine, where they added the new adjuvant right before it was released, and they said that it had been studied thoroughly, but it actually was a slightly altered vaccine before it was rolled out to the public. And
that's the swine flu vaccine from 2009.
Exactly.
Right. So, at least in the UK, people were, presumably there was a public notification that the vaccine that was tested was not the one that was being given. But here there was no notification. Nobody knew about this. I mean,
that's exactly right. But they knew. I mean, the FDA knew, and Pfizer knew, and the FDA even knew that there was lower mRNA integrity. And they knew enough. They knew enough to ask for these 252 extra participants. And so they knew. So Pfizer had disclosed that there was this new process that they were using, right, to make the vaccines. And now we know, looking back too, that there was actually, it was a 2.4 times higher rate of adverse events actually among those 250 who got is that right? I saw an analysis of that. Right. They were mild, like side effects, basically. And so we knew that they were mild. So at least if they had done some preliminary analysis, they would have seen that. They didn't. They granted the EUA, I guess, before they had that information. But then, so now, like, so
can
what
I just
happened,
interrupt again? So there wasn't an analysis of the side effects of this process 2 batch compared with process 1 batch. Is that something
I've done? So Retsef and I have done that analysis. It's a little bit more nuanced than that. But if you just we didn't do the comparison that they anticipated, which was to take a random sample of 250 people and compare. I mean, we could do that. There are different ways you could do that. In the most basic analysis, where you just take everybody who was in the treatment group, the treatment subjects who had gotten process 1, and you compare their adverse event rate to the adverse event rate of the 250, there's an increase about 2.5 times the increase. Now, when they recruited those 252 people, they also recruited placebo subjects at the same time at the same sites. And if you do that comparison, you see about a similar rate of increase in, the process 2 subjects have about a 2.4 times the adverse event rate than the placebo subjects who were recruited there. However, in our analysis, one of the problems with the comparison that we're making is that different sites at different times, all of the subjects had different adverse event rates. So if you take a comparison, so even the placebo subjects at those sites at that time have a higher adverse event rate than other placebo subjects at other sites at other times. So in some versions of the analysis, we don't need to get too deep in the weeds. We don't see with the sort of the increase in the subjects and the treatment subjects at those sites at that time is similar to the increase that the placebo subjects also got at that site at that time. Now, why are those? Why is there so much variation across sites and time periods in the adverse event rate reporting? This is a really interesting question.
So
I think the cleanest what?
Yeah, you did publish this. We can link to that as well.
No, I actually haven't. We haven't published it.
Okay. There was an analysis done on Daily Clout that we could link to, and you
could link to that. That's the one that does the comparison between, and that's the cleanest comparison. Is you take the same sites at the same time. You look at the placebo subjects that they recruited at the same time that they were recruiting those 250 to treatment subjects. That's the cleanest comparison. Now, I think that if you were to dig into that, you might say, well, a lot of these adverse events that are being reported by the treatment subjects at that time could be called, reactogenicity, which we all know there's a very reactogenic vaccine. So, you know, I mean, there may be different ways you could try to dismiss that. I think what you can't really dismiss are some of the very clear safety signals that emerged after the clinical trials that didn't see in the clinical trials at all. And that can, in many, you know, mechanistically be connected to the process to manufacturing method. So one of the clearest ones is lymphadenopathy, okay, swollen lymph nodes. In the trials, process 1, process 2, the rate of lymphadenopathy was about 0.4%, okay? Later, when they trialed the booster using process 2 doses, as far as we know, the lymphadenopathy rate shot up to about 5%. So you have like this huge jump. And one of the, you might say, well, that's because it's the booster dose and not the first dose. But we actually know from very careful PET imaging studies done in Israel that there was no increase in the lymphadenopathy rate. And this is very carefully measured, right? No increase in lymphadenopathy from doses 1 and 2 to dose 3, right? The first booster. So it's not a result of a booster. It's a result, I would argue, of this switch from process 1 to process 2. So that's one very clear signal. Another very clear signal, in my opinion, is the menstruation problems and the vaginal bleeding that we're seeing now. This emerged very early on, right? Many women reported this. And initially, it was dismissed as being due to stress or, you know, women being hysterical or whatever. I say that tongue in cheek. I mean, I don't think they're hysterical, but that was kind of the zeitgeist at the time. Or, you know, but, you know, okay, we should study it further. Well, we know now, I mean, there was just a publication in Science Advances looking at this, showing that women that don't normally menstruate, mainly because they're on some kind of contraception, about 7% of premenopausal women experience vaginal bleeding within a month after vaccination from the COVID mRNA vaccine. I don't know if it was just mRNA. Yeah, no, I think it was the Pfizer vaccine specifically had this rate of 7%. Mm-hmm. And so what was the rate of, you know, reported in the trial? It was 0.7%. And that's, again, premenopausal women. So how do you go from 0.7% to 7%? Well, either you're doing a terrible job of tracking adverse events, in which case the whole, you know, trial, validity of the whole trial is in question, or it's because you've introduced a totally different product, and we can talk about the different mechanisms that might trigger menstrual bleeding. So that's another one, and I would argue anaphylaxis is another one. But.
And the important, I just want to highlight one important aspect in what Josh saying, that the rates of these newly discovered adverse events, the observed rates in practice are high enough that you should have expected to see a significant signal in 20,000 people. So it's not like one in, you know, 100,000 that you can say, okay, I ran the trial and I couldn't see it. Like 7%, you expect to see something that is more than 0.7% in a 20,000.
So just to sum up, we have this situation where we have a lot of indications that something important may have changed from process 1 batches to process 2 batches in terms of their adverse events. And that is due to the differences between the 250 people, a very small number that was investigated as part of the phase 3 trial, but it's also because we see a different rate of these side effects post-marketing than what was seen in the phase 3 trials, which of course all of this is subject to criticism if you don't like the results, which will be about, well, increased awareness and things happen. There are a lot of things that can be explained as coincidences, which I think we also touched upon early on, that we're just having so many coincidences. So it's getting somewhat hard to maintain that this is.
The other point, though, I think it's very important to emphasize this. The onus of proof, of the burden of proof, is on the regulatory agency.
Absolutely.
And we talked about history a little bit, but this whole issue around the manufacturing process being a major aspect of the regulatory process and the approval emerged after the Qatar incident, where basically the polio vaccines, exactly same scenario, by the way, were tried in a major, like huge trial, but the manufacturing process was highly controlled, and then they were trying to scale it, and they didn't do it right, and they used different, somewhat different process, and they ended up infecting, many children with polio, because they did not make sure that the polio virus in the vaccines was actually dead, as it was supposed to kill, as it's supposed to be. So I think that people need to understand this is not just like something minor that they kind of a small corner that they kind of rounded. This is going against fundamental, based on hard lessons from major safety incidents that we had in the past. And to some extent, we suddenly ignored everything we know. And again, as you said, like now you have to suddenly prove that something is unsafe.
Yes,
which
and
is,
I think that's
not what
you talk about the bias that might be with regulators and health authorities in general who were promoting the vaccines, but it leaves this vacuum, which I think is I came to think about when you talked about it, just that now an independent researcher from Israel is now trying to pull this data out and do a decent analysis, and your job is so difficult, and I don't think. I think the general public maybe sometimes perceive that we can all access this data, and why can't we just study this if the regulators don't do? But the problem is that those who have the good data are the regulators, and they're the ones who should do their job. And it's extremely difficult to come in from the side and get access to the data, if at all possible. And also your ability to do these comparisons, as you talked about, Josh, are hampered by the fact that you don't have the full data set. And we could do so much more as researchers if we had access to all this data, and actually really in real time, at the same time as the regulatory bodies could be analyzing and chipping in with our analysis, and if we thought that something wasn't covered well by regulators. I think that would make for a much more safe system, because it's really a strange process now that we are in this situation where you have to sit there and do the best you can, and I think you have done that with the available data, but your hands are somewhat tied by the fact that you don't have the full access.
And this is a very unusual circumstance that we even have access to the data that we do, right? Because this was brought about as a part of a court action against the FDA, where the FDA didn't want to give this data through a regular FOIA request, so they had to go and basically sue the FDA for this data. They wanted to give it out, they wanted to kind of trickle it out over the course of 75 years. Thankfully, the judge interceded and forced them to release it earlier. Yeah, it would be great if we had access to the data, on an ongoing regular basis. There's kind of no reason why not. One thing that's interesting is that, another part of this is that the regulators rely on the manufacturer to provide them with all of this stuff, right? All of the data, all of the comparisons, and all of the assays and tests that are chosen to do these measurements, and there are problems with that, are chosen and by the manufacturer to kind of present the best picture that they can. And it was never confirmed by the FDA. They didn't do their own independent testing.
Yeah.
So maybe we should move on.
Christine, the point you made, I think, is relevant not only to this vaccine. I think it's a much more general principle that we should seek to, right? Like that there will be a transparency that allow the scientific community to do the research and see, if they see something that the regulatory agency did not see.
That they shouldn't want people to have access to that if what they really care about is like the safety and efficacy and having accurate results. They should want more people to be able to analyze it to help.
Right.
But and I have to bring up something else that's like I've been thinking about as we've been talking, is that we do have more data than we've been discussing. And I don't know if you've looked into this, Josh, but I mean, we have the trials in the adolescents, And we have the trials in the 5 to 11-year-olds and the under 5-year-olds beyond that of the booster. So those would have all been process 2. And so we
presumably.
Well, the
presumably.
adolescent trial used both process 1 and process 2.
I'm glad
There
I
was
asked about
actually
that.
only one site in the trial that used process 2. No, that's not true, but one site that we know of that used process 2 doses. We don't have the full data on that because it hasn't been fully, it's just starting to be released now.
So the other ones were process 2, but the adolescents, it was like only partially process 2. And so we could get some answers from that if we had the data in terms of looking at safety, but it's so small.
We are looking into the
Small.
phase 3 trials right now, and
Yes.
summarizing in a review we'll be submitting just in a few weeks about the differences in adverse events in the different age groups. And I think we see a very strong trend with age, which is probably linked to the dose, because they lowered the dose of mRNA in the vaccines in the younger age group. So that comparison is already
right. It is problematic. Yep, definitely. Yeah. That's
the 12 to 15. The 12 to 15 is still at the 30 milligrams. So that might be.
Exactly. So we could use the 12 to 15.
Yeah, the other thing, the other thing that I think is the other thing I think is interesting here, that I'm actually not sure that there is only process 1 and process 2 period. I actually suspect that there could have been also modifications to process 2. At least we know, at least on one of them, when they changed the adjuvant as well, like they added. So I actually think that there is a broader and deeper lack of transparency about the changes in the manufacturing process and how they evolved over time, and I'm not sure it's just process 1 versus process 2 period.
Yeah,
right?
and a good indication of that is from a Pfizer clinical trial report, where they showed that there was a higher adverse event rate, serious adverse event rate, and adverse event rate of special interest in the placebo group that crossed over and got vaccinated, which was at the time of the report was almost like it was over 80% of them. And they wrote in the report, it was as expected, it's higher. And then the question is, well, why are you expecting that to be higher? We know that, there seems to be some indication of differences in some of the lots that were sent to the trial sites at the period when they would have been giving these doses to the placebo group. Again, the data doesn't allow us to exactly identify which lots were given to which people, except for that 252, we can identify those. But it raises a big question. We published a rapid response in the British Medical Journal, pointing to this issue of, we don't know how the variability in manufacturing processes affects the safety and efficacy of these vaccines. It's very understudied, and this is information that the company is holding very close to its chest.
And I think a lot of us.
Can I give you an example?
Yeah,
I'll give you an example. Okay, we know that the trial. Maddie DeGarry. So Maddie DeGarry was a 12-year-old girl who was in the trial, and she suffered horrible, debilitating injuries that appear by any metric to have been caused by the vaccine, but the manufacturer and the site investigator refused to acknowledge that link. In any case, she was at one of the sites that we know that was given the process 2 doses that were used as part of the adolescent trial. We don't know if she got that, and then her parents have asked for the vaccine card, right? The proof of vaccination, and on the vaccine card, there's a number that's written for the batch that was given to her, but that number is a generic number that applies to all of the batches and all of the lots. And so she hasn't been given specific information about what lot she was given to. That's how closely this information. Now, all of us that went out and got vaccinated, if you got vaccinated, you were given a card, and it was written exactly. You could identify which lot, or at least which batch, you were given. But the information she was given was generic.
And it reminds us, even the people in the trial didn't have informed consent. You know, we talk about how the general public didn't have informed consent, but even the people in the trial who got process 2 didn't know what they were getting. And I think maybe that this is like a good time now to talk about what exactly, sorry, I think I froze there. What exactly is the difference that we know of in the components of process 2, or how the manufacturing exactly is different, and you know, why are we worried about this? I think we should bring the listeners up to speed.
Sure. Well, you guys just did a study, a podcast on the DNA contamination, right? So this is exactly related to that, okay? Because what do you need to make RNA, mRNA? You need DNA. You need a template, right? And the way they did it with process 1 to create so you need a lot of DNA to make a lot of RNA. So the way they did it in process 1 was they basically used PCR, which replicates nucleic acids, right? DNA. So you put it in a PCR and it replicates this. In the process 2, they used E. coli bacteria, and they put plasmid DNA inside of the bacteria, and then they sort of had the bacteria replicate. And when the bacteria replicated, the plasmids also did. So then they have this template, and then they have to basically extract that template from the bacteria, try to purify it as much as possible, and then use that template to make the mRNA. And there are all kinds of additional problems and risks that are introduced when you start using E. coli bacteria. So that's really the key. And there were also some differences to the lipid nanoparticle manufacturing process, as far as Pfizer has announced. So that's really the key difference. And so now what we've discovered, right, is that there's large amounts of DNA fragments from the template that are still in the vaccines. And in addition, there's the membranes of the E. coli bacteria, which are called lipopolysaccharides, otherwise known as endotoxin, which are highly inflammatory and highly toxic. More toxic than ricin, I think, was something that I saw recently. So then the question is, well, how much is in it, and is it dangerous, right? And so if you look at the analytical comparisons that were presented to the regulators, all of it looks like to be within regulatory limits. Now, why are those regulatory limits, set at what they're set? I guess I don't know the full backstory behind that. But what I do know is that this is a new technology, right? And one of the key differences is that you have these lipid nanoparticles where the contents of that are transfected into cells. Now, what we're interested in transfecting is the mRNA, right? But what gets into those lipid nanoparticles, you also get the DNA fragments. You also get the endotoxin. And so whatever, regulatory standards were set based on a kind of regular injection or biological injection don't apply here. They can't apply, right? And there doesn't seem to have been any effort to update and do any real testing on, well, okay, now that we've got this new technology, how do we need to change our standards? Another problem is that again, they let the manufacturer decide what tests to do and to do the tests. And so the tests that were chosen for looking at the DNA contamination will miss small strands of DNA, okay? And so they basically missed a huge proportion of how much DNA contamination was really in the vaccine.
But even what is detected, at least for Pfizer, is above the limits, right? So I think.
Well, no, not what the regulators, not what was presented to the regulators. What was presented to the regulators was
the external.
Right.
The
external tests on it have shown. I'm not sure about that.
I think for Pfizer, I'm pretty sure that for Pfizer, at least.
Yes, well, they're all over the limits. And every vial they've tested basically has been over the limits. But if you use what the method that Pfizer used, you won't necessarily detect it as being.
So they weren't necessarily detecting all the DNA and the fragmented DNA and the endotoxin because they were encapsulated, right? So there's another
both
method.
because they were encapsulated, but also for the DNA because the method they were using was undercounting small fragments. So there's
and I think the rationale was, I think, that what you're really worried about are long sequences of long fragments of the DNA. That's kind of more concerning because that could have more meaningful transfecting into the cell. But I think that major experts in the area now actually claim that potentially actually the smaller fragments are more risky because they have a better chance to potentially integrate themselves into the DNA of the cell. And now, once that happens, now you have cells that have broken DNA or malfunctioning DNA, and they replicate it. That could lead to, very bad. At least theoretically speaking, it could lead to, cancer and other bad, autoimmune reactions and stuff like that. So I think that there was potentially rushing to make an assumption, and I will relate to what Joe said, that when you rush a new technology and you then make also very, very kind of relaxed assumptions, that's not a very good recipe for safety. Let me put it this way. And again, I think we need to distinguish between: am I going to approve it for emergency use to high-risk people? Okay, maybe yes, because with all. But am I going to map all of these uncertainties and continue to scrutinize them and check? That had to be done, and I don't think it was done. And that's kind of, to me, the biggest process level issue. I think there are many concerning findings, but I think one of the things that maybe is someone that looks a lot of safety and system level safety. To me, you have to ask yourself, what is the quality of the process? Are you asking the right questions? Are you doing the right test? Are you having the right checks and balances? And I think when you look at this, it's very obvious that was all thrown out of the window. And I don't think there is any excuse for that when it comes to the low risk younger population. You may have an excuse. Okay, I had to rush for the high risk population, but even then, you had to do that with full transparency and sort of really have an informed consent that is very transparent. And unfortunately, neither happened. It wasn't any informed consent, but also they rushed it also for the young and the young people and children, which I think it's very, very wrong decision.
And
we
maybe
still
I
have
can
the FDA, saying that it's emergency for under 12-year-olds because it's still under emergency use authorization in the U.S. And that's, of course, decided by one person, Javier Becerra, that it's still an emergency. And so it's really crazy to think that it's still, considered an emergency in that age group. And one more thing I wanted to say was the other thing that has come up as a concern is that, and in the DNA of the Pfizer vaccine, that Ontario study, they just kind of elucidated that there's the SV40 promoter, which is simian virus 40, which has been known to be like tumorogenic, the entire virus. And I don't know, I don't have the expertise to say how, like if it's oncogenic or can cause tumors, the promoter itself, but I know that it being there has been a concern to many people. And it's just another example of a question that we don't have an answer to, and highlights, the fact that we didn't have clinical trials. We've had all observational data since the original trials.
Yeah.
But yeah, you guys have things to add.
I just want to add something to that. So Pfizer presents their plasmid template, and the components of it to the regulators, and they have this promoter in it, and they although they presented the full template, they're supposed to point out what are the different elements of the template, and they didn't notify any of the regulators that they had this SV40 promoter in the vaccine. So that's a huge regulatory lapse. What?
So now is Pfizer liable because they didn't disclose that?
People are starting to argue that yes, because there was this contamination that wasn't, contamination isn't the right word, I can't think of the right word, but now because it was in there and it wasn't disclosed to regulators, yes, that Pfizer may now be sued. I don't know if that's the case. I don't know.
It's called, yeah.
And it's important to, I think, underscore the fact that, I've been working in the regulatory space of manufacturing biologic drugs, for 8 years now. And so by now I kind of seen a lot of stuff and talked to a lot of people in the industry. Any one of these things would be immediate stop. like if you have a contamination of that level and magnitude in your product, impurity, that's just like you stop everything, and you need to scrutinize the manufacturing process, and you need to really show that you are able to remove that impurity. It's clearly violating any limits that the regulatory agency did. So either the agency has to explain why these regulatory limits don't apply here, or they just simply have to stop. And what we see is quite the opposite of that, right? We see that they continue to push this through, including to young people,
Yes.
including to babies, and that's
Yes.
it's like
Is it because it's a
Until-
militarized process, and hasn't been the standard process because it was an emergency? That's what I'm trying to figure out, how much we need to worry about this for future vaccines.
I think we have to worry about this. Well,
well, I mean, the notion of regulatory capture is a standard concept in political science. It's not new. It's not conspiracy theory, right? The idea that regulators come to kind of serve the interests that they're supposed to be regulating, hurting the regulatory duties, is commonplace, right? It's not an old thing. So you combine that with what Retsef was saying, where they're basically cheerleading this and requiring people to take it in order to keep their jobs and all this stuff. And then it turns out that they did a terrible job. The last thing they want to do is admit their mistakes. It's interesting though, because until now, all the regulatory agencies have ignored this issue of the DNA contamination until I believe this last week, when the regulator in Canada acknowledged that there was this contamination, or at least they acknowledged the SV40 promoter, but they said it's business as usual. It didn't matter, and
nothing to worry about. It's safe and effective.
Nothing to worry about.
When did they learn about it, though? That's the question. Does anyone have the answer to that question?
So this is the problem. I think we've been discussing. Okay, what would you want to do, right? Okay, one of the risks, for example, the theoretical risk, that it's not completely theoretical, if I would say, because we've seen that in vitro and in other contexts of DNA-based treatments, that you can really have an integration of fragments of DNA into the DNA of the cell. Right now, one natural idea would be, hey, take people especially people that were harmed from the vaccine, and test their cells. Take people that maybe develop cancer shortly after the vaccine and test their cells. That this is something doable, that you would expect that it will be initiated by the government, and at the very least allocate funding to do that. I think that I suspect that if someone will try to do this type of research, well, first of all, logistically, it's not obvious, right? Like, you need to have access to patients, you need an IRB, you need funding. I think that the chances, and of course later on, the scrutiny that you will get if you find something, it's almost like creating an environment that has all the incentives not to touch it for most researchers, because they can just, what do they have to gain here other than, so I think that this is another striking example of how we actually live in a world where not only there is no transparency, not only that the regulatory agencies do not do their job, but in fact, there is an environment where people are the research community, scientific community, is basically implicitly and explicitly are being sent the message, just don't ask questions, move on.
Right. Well, there are huge costs, based on the research that we did. There's huge costs to researchers who try to raise concerns about the risks of vaccines, and you, Christine, probably are familiar with with that as well as anybody. But the other thing is that regulators and agencies like the CDC are very hesitant to acknowledge risks of vaccines because they're worried. Like, so for example, if now they discover that there's this problem and they admit that there's this problem, there's a broader concern that it will hurt people's confidence in vaccines more broadly. And this idea of guarding against doing anything to guard against vaccine hesitancy is a very, I mean, I can understand the logic behind it, but I think that it's come at such a huge cost to protect the vaccine program, to guard against vaccine hesitancy by really repressing and rejecting, scientific evidence of vaccine harms is a problematic attitude, and especially now that they're trying to, they want to create a whole bunch of new vaccines based on this technology. And wait a second, now if we acknowledge this problem, it's not just about the COVID vaccines, it's about all of these COVID vaccines, and there's billions of dollars that's been invested into this technology, and so there are huge vested interests that, in maintaining the legitimacy of the mRNA platform.
But empirically, actually, we see the opposite reaction of what they're trying to prevent. Like, in fact, we see vaccine hesitancy is increasing because of the lack of transparency in Israel.
Yes.
By the way, Israel is a striking example. I don't know if you're aware of that. So Israel doesn't have vaccine mandates, and typically for traditional vaccines, the adherence rate was north of 90%, and often actually north of 95, sometimes 98%. I think actually the COVID-19 vaccine was the only time in Israel where we had pressure. I mean, people would say it wasn't a pure mandate, but for many workplaces, it was a mandate, and it was like green passports and things like that. But the first two doses, the adherence rate was 86%, 80, close to 90, definitely the high risk population. And the booster, it went down to 50%. And then the more and more boosters, it's just like really at the level of fractional percentages. But now we also see a decrease in the adherence to other vaccines. So I think that what they're accomplishing is exactly the opposite of what they are trying to prevent. They are just actually, I think they opened a can of worms, because now people are asking questions about all vaccines, because once you saw a bad example. I can tell you personally, I never doubted any vaccine. I took all the vaccines. I gave my kids all the vaccines. It's much more similar to the US schedule. I took the two first Moderna doses. I personally, as a scientist, I'm not accepting anything now. I'm just saying I want to see it in my own eyes and scrutinize this. And once you start to do that, you see some strange things, like why a baby at day one should get a hepatitis B. Like, it doesn't make any whatsoever. Like, or why would a baby get like 8 vaccines on the same day? Like, that doesn't make sense. And I think that they have to change course, because I don't think they're going to be able to address that with saying, "Hell, safe and effective. This doesn't work anymore." I don't think it's going to work. And I wish, because I think that the damage is not going to stay for vaccines. I think that fundamentally we are hurting the trust between medical professionals and their patients, because patients cannot. By the way, I think that many doctors are not even aware of all of this stuff. You will be shocked. I talked with some physicians that I actually consider very good physicians, including my own personal one. And when I started to say, hey, that there is myocarditis, yeah, but all of this research, and they were like, what? Like, and they asked to see, actually, to their credit, they asked to see. But I think that this is not going to stop. And I think regulatory agencies, if they don't switch their kind of flip the record and like change their mindset and their approach, I think they're going to face an increasing problem here. It's not going to go away, in my opinion.
I hope you're right that things will change, but for the time being, I think we are stuck with a system where it's the manufacturers and regulators who have the access to the data and can analyze it, and they sit on it, so independent researchers can't get access. And we also touched upon all the biases that are against I'm really happy now. I have a postdoc who has entered into the field of looking at the effect of COVID-19 vaccines to Danish children on overall health, because I know many people will be discouraged, many young medical doctors will be discouraged from entering this area of at all just opening data sets to look for effects of vaccines, because you face so much adversity, or you might face a lot of adversity if you come out with results that aren't just completely in line with what was anticipated. So we have all these biases. At the same time, we have the situation where you guys have uncovered that we have a major problem, that we are now vaccinating people with a vaccine that was never scrutinized in the way it should have been, because we are vaccinating with process 2 vaccines that were never under investigation in the phase 3 trials.
Christine, your microphone is echoing. Do you guys hear that too, or is that just me?
Yes, I do hear.
Yeah, okay. Because I could tell Retsef heard it too. I was like, it's like a double of your voice. But I want you to continue.
I don't know how that happened. It might be something to do with your connection.
Maybe you have it on open in 2 places.
Is it better now? No, I don't.
No. You might consider disconnecting and reconnecting to the stream. It might help, maybe.
Since I'm doing the recording, I don't think that is so good. So I'll
okay.
I'll stay silent and let you guys do the talking. You were saying great stuff anyway. but if you understand one question, then that would be where do you go from now? How can we move on in the current situation? To answer that question that I know listeners will have, how can we get more information about process 2 and the risk associated with DNA contamination and endotoxin and so on?
Great question. I think I feel like we're not going to make any headway with the regulators. I've just seen such really atrocious behavior on their part, on so many levels, many stories. Anyway, I think maybe one way forward is legal, legal proceedings. Now, hopefully, this revelation about the SV40 promoter will crack things open. And there are many, many legal cases moving forward in various jurisdictions around the world. And people point to legal protections in the US that make it nearly impossible to sue the manufacturers. And although they were given indemnity in other locations, the laws are different and the protections are different. And so, if we can crack the legal barrier and get discovery, because a lot of the data that we would want is not part of the release that was made of the data. I think that, hopefully legal liability and the transparency that comes with legal cases will hopefully provide us with a good, way to whack at them.
My sense, a few thoughts about this. So first, I think that I suspect that in the US, if we want to do research like that, we will need to find some private people or organizations that will put this budget, the funding to do that. I actually am more hopeful that this type of research will emerge from actually the Scandinavian countries, if I may say, because I think that so far they've been, relatively speaking, more open and more kind of allowing the research to be done. But there is another approach that potentially can be a first step. So one of the challenges here is that, okay, to do large scale or just even if you have the funding, you need an IRB, and like IRB is being controlled by universities and academic institutions and other hospital systems that potentially are part of the problem. But I wonder if someone will develop some diagnostic kit that can be available commercially, and then people that were harmed can apply and just test themselves, right? And if they find that there is an impact, they may be able to then use that in the public domain or legal level. Now, it's not completely obvious how you do that, but I think that there are potentially some legal paths to be able to do that. Which, I mean, it's not clear yet if it's a viable way forward or not. I think Josh and I have heard different opinions about that, but
right.
but it might be something to consider. So.
Two responses to that. One is that they are developing a kit. They are developing a test. They, meaning Kevin McKernan, who first discovered this, and some of the people that he's been working with. So hopefully that'll be available soon. And the other is that there is now the Institute for Pure and Applied Knowledge, run by James Lyons Weiler, has started an independent IRB body. I'm actually a member of it. And that's going to get certification soon, and then that would create a pathway for people to get the IRB approval for these types of studies.
That's great. I think we need to keep pushing forward because, frankly speaking, I think that if you are for medicine and if you are for vaccines, as at least, I think we all believe that there are some vaccines that are doing good, have good outcomes. I think that if you actually are in favor of medicine and vaccine and specifically, you have to actually be at the front line of this fight, because I believe that what they are going to do if we continue on this path, they're going to destroy any trust, and without trust, I think there's very little chance that you're going to convince people to take, in the future. Definitely
right.
Vaccines, and the reason why vaccines are more sensitive to lack of trust is, unlike other drugs, you have to take vaccines when you're healthy. So, like upfront, like why would I take? If you think about it, why would I take something if I'm healthy? That's a much less obvious situation. When if I have, let's say, God forbid, like advanced cancer, and like you offer me, you either die or you try this drug. Okay, probably much more convincing proposition. But like vaccines we give to not only to healthy people, to young, often young and children, young people, children. Like, why would someone want to take the risk on that unless they are absolutely sure that not only there was a lot of scrutiny to even approve that, but also a lot of scrutiny going forward to make sure that if something is wrong, we stop it and or at least address that. So I just think that we need to be on the front line if we really care about medicine on trying to make sure that we are taking a different path.
I totally agree with that, Retsef. I just want to say, I think the way forward too is beyond legal action, which I know that's necessary. That needs to be part of the process, is independent researchers really probably needing to create a network, multiple networks with each other about where there's funding, which universities are kind of leading the way and supporting this sort of research. And, and so the more people, the more momentum that there is with, creating, independent research and asking for transparency and validating other, groups' findings. And the less fringe it is, the less we appear to be like the anti-vaxxers, but more like the pro-science, people in the actually pro
safety,
health, pro-safety group. Then I think the more traction the movement gets, then there'll be a real challenge to our regulatory agencies, because the public is going to be turning to independent scientists, as they have been doing, to get information that they can trust. And, right now, I mean, we're in a tough situation because scientists are like losing their jobs. They can't get funding. They're, being publicly humiliated and called all sorts of names. But I think it's kind of up to all of us, and also the people who are at universities and heads of universities, to say, this isn't how science works. Science works by, allowing differing voices to, have a say, and to allow questions to be asked and the studies to be done. But speaking as someone who's worked in the US and Denmark, I mean, getting private funding in Denmark for research, I thought was a lot easier. It's sort of more built into the system. Whereas in the US, so much of the medical funding is based on NIH. Yeah, Christine's not so sure. I don't know, about half my PhD was private funding and half was public, but and so I know it's possible to get public. I mean, to get
There's a
private.
difference between possible and easy. I think that's what Christine is saying. It's maybe
Right,
not as
yeah.
hard as getting a
Probably depends on what you're studying, actually, but yeah.
And how much money you need.
Yeah.
Funding a PhD student is different than a.
Well, you know, I had yeah, I guess I raised almost $1 million because I ran a
Okay.
very large study and
That's
had to
impressive.
employ 6 other people. So yeah.
Okay, all right, I give it to you. All right.
okay.
So can you hear me now, or am I still echoing?
A little
You're
bit,
still echoing, but
that's fine.
we can. I mean, we can understand you. It's just a little,
Okay.
little off.
Well, anyway, I just wanted to hand over to Tracy maybe to wrap up, since we've been talking for 1.5 hours now. I think we've touched a lot of ground. I'm thrilled by this discussion. I think it's great to be with you, and I've just felt like clapping all the way through about the things you have said, and how how we are all in favor of vaccine safety, and that is achieved by presenting the public with data and being transparent about it. That is really how we build trust in vaccines in the long end. So I think we touched upon very important things, and also some things we can't solve right now, which is about how do we move on. We don't know the clinical implications of the process 2 yet, but we need to move on to understand better what it implies clinically, and we need to find that path to do so. And I was encouraged to hear that there are tests underway. We are also right now in Denmark looking into how to do a good investigative immunological program for people both with long COVID and also with complex diseases, multiple organ diseases after vaccination. There are many similarities, and I think we need to investigate more about the immunological pathways to be able to come up with good diagnostics, but also good treatments, and in the future to be able to prevent vaccine adverse events clearly. But anyway, Tracy, I'll leave it to you to wrap up, or maybe ask your final questions. And here, if you have final comments, Josh and Retsef, it's been so great to hear from you.
Yeah, I agree.
Thank you for inviting us. So it was a pleasure and interesting.
Yeah, it was a great conversation. Just really appreciate you guys taking the time to answer our questions. So I think we'll leave it at that.
Enjoy the weekend, everybody, the rest of the weekend. Take care.